Video we're going to look at resource acquisition. So how plants get resources, and transport. How plants distribute resources. So basically how plants take resources from the ground or their surrounding environment and distribute it amongst all the cells that make up that plant. So there's both long and short distance transport, and the first thing I'm going to talk about is short distance plant transport. So plants must move resources they need, like minerals, water, sugar, carbon dioxide, and nitrogen, into all of the cells that make up that plant, and get rid of waste products like oxygen from every cell in the plant. So places that need water, sugar, carbon dioxide, specifically those areas that photosynthesize, will need to be constantly supplied with those. And those same places that photosynthesize will be getting rid of a lot of oxygen into the environment at the same time. On the small scale, so from cell to cell, this transport usually happens by osmosis and diffusion. So things like carbon dioxide diffuse into a cell, or water diffuse into a cell, waste products like oxygen diffuse out. Sometimes, especially on hot days, plant cells need more water water at a faster rate than can be supplied by osmosis. And so they have these facilitatory proteins in their plasma membrane called aquaporins that facilitate the natural osmosis of water into and out of the cell, just to make sure that it can stay up with demand. Like we talked about in the cells unit, water potential directs the flow of osmosis in these cells, so from areas of greater water potential to more negative water potential. However, in plant cells, we've got this rigid cell wall that can provide positive pressure against the inflowing water. So we can actually get a positive pressure potential. Once water starts to push against the cell wall can push back, and if that positive pressure provided by the cell wall is completely equal but opposite to the solid potential, we'll reach equilibria. So materials may travel from cell to cell, osmosis diffusion, by two routes. The apoplastic route, which is kind of this green area, traversing through the cell wall. Apoplastic route. See, apoplastic route through the cell wall, through the cell walls connecting those cells. Or the symplastic route, going completely through the cytoplasm or interior of one cell and through these channels that hook together the cytoplasm of two neighboring plant cells. These are called plasmodesmata. Plasmodesmata. So two ways that we can traverse between cell to cell in plant cells. So if we put a plant cell in a hypertonic solution, water will start to leave through the aquaporins, and the interior portion of the cell will actually peel away from the cell wall. This is called a plasmolysed cell. If it's in an isotonic, water in and water out, the rate is equal. These are flaccid cells. And if more water goes in a hypotonic solution, it makes a very stiff, bursting cell. That's a turgid cell. And so flowers wilt because they lose more water, evaporate more water, and all of their cells are plasmolysed. A nice, freshly watered, plump-looking, great-looking plant is turgid because its cells got flooded with a hypotonic solution of pure water. Plant nutrition depends on symbiotic relationships. So many organic and inorganic resources that a plant needs, like nitrogen, minerals, phosphates, etc., come to the plant from the soil via the root hairs. So the nutrients travel into the root via the root hairs and then into the xylem and phloem and up through the plant. Plants must rely on other living organisms in the soil to replenish the soil supply of needed nutrients, like nitrogen. Nitrogen exists in the air as N2 gas. N2 gas cannot be used by living organisms in that form. However, plants can use nitrates and ammonia. So bacteria in the soil turn N2 gas into ammonia and nitrates, those forms that the plants can use. And this is called fixing, or nitrogen fixing. So nitrogen fixing bacteria in the soil can turn gaseous nitrogen into ammonia, and nitrifying bacteria can turn that ammonia into nitrates, which then nitrates can be absorbed into the root system. Ammonia can be absorbed into the root system, and actually ammonifying bacteria can turn rotten organic material into ammonia. So 3 sources of nitrogen that can be taken up by plants, relying on this symbiotic relationship with bacteria. Also, plants can rely on mycorrhizae, which are mutualistic fungi that lives on the root tissue. See, here are these fungi that are living on the root tissue. These mutualistic fungi help provide the plants with minerals, phosphate, and nitrogen, and in exchange they consume some of the sugar produced by the plant and stored in the roots. So it's a symbiotic relationship, mutualistic relationship between mycorrhizae and these plant roots. A lot of nitrogen-rich or nitrogen-limiting plants like orchids have an extensive mycorrhizal network. Peanuts do as well. So transpiration draws water up into plants. Water and minerals enters the plant through root hairs and travels via the apoplastic and symplastic routes towards the xylem. So minerals and water travel in through the root hairs, traveling via apoplastic red and symplastic route in blue, and then it gets really close to the xylem elements or vessels and passes what's called the endodermis, so a bunch of cells protecting the xylem, and they must pass this Casparian strip, and this is a strip present in the cell walls of these endodermal cells in the cell wall that are waxy, and they prevent any material passing through or water passing through the apoplastic route. So once this water hits the Casparian strip, it must switch to the symplastic route and then finally be pulled up into the xylem, where bulk flow of water and minerals through the xylem distribute it from the roots to all the upper cells in the plant. This transport happens because of transpiration, and transpiration is the evaporation of large amounts of water from the leaf tissue. So leaves are hanging out there in the sun. They require a lot of water because they're going through photosynthesis to split that water for photosystem 2. And that water evaporates. It gets lost into the atmosphere. This transport is possible because of that water is adhesive and cohesive with each other. Water as transpirated or evaporated off of the leaves is via hydrogen bonds cohesively bound to the water molecules below it. And so when it gets pulled out in the atmosphere via evaporation, it pulls the next water up through the xylem into the leaf tissue. This pulling action creates a negative pressure or tension, which can be traced all the way down to the roots. So this pull from the leaves can work as a large, almost column of water pulling the next and the next and the next and the next molecule of water through the xylem all the way down to the root tissue. And this is called the cohesive tension or column of water hypothesis. So here's a picture of how that's happening. When water is absorbed at the bottom in the roots, the pull of that water from the leaf tissue through the stomates into the air, this transpiration, pulls the next water molecule up, pulls the next water molecule up along with it. So all of these molecules of water are being pulled up through the xylem via cohesion to the next water molecule above it and adhesion to the water molecules to the xylem elements, the tracheal elements of the xylem, all the way up from the root. And so what you'll see is if we track water potential, the pressure potential, or the water potential, is very negative, the outside air very negative at the top and less so negative at the bottom. So water potential is gradient, it's increasing water potential more negative towards the top, and so water will flow through the xylem towards the top, even through huge trees like in redwood forests. On hot days, in fact, during the summer, when water is being lost much faster than it can pull water up through the roots, you'll see the tree diameters will actually noticeably decrease due to rapid transpiration, because such a negative water potential is being formed in here. It acts like a vacuum and sucks in all of the vessel elements for the xylem. You should measure it sometime or go out in the forest and look. It's actually noticeable. Stomates or stomata are actually what regulate the transpiration rate. So water is lost through plant leaves through these tiny openings called stomata. They kind of look like this. These are all they look like lips almost. They're stomata, and they must open to allow carbon dioxide into the leaf and to let water and oxygen out of the leaf. Most of a plant's water, like 95% of it, is lost through these stomata being evaporated into the outside atmosphere. These specific lip-shaped looking things that border the stomata opening are called guard cells, and they open and close to regulate the amount of water that's being lost or carbon dioxide needs to be taken in. Stomata are usually closed during the night when not a lot of photosynthesis is happening to limit water loss or transpiration, and open during the day to maximize CO2 influx into the leaf tissue to maximize photosynthetic rate. Desert plants have lower stomatal density, so less stomata per unit area on the plant leaf, than, say, marsh plants, because they live in a very hot, dry climate. If they have more stomata, that's more water that can leave, and pretty soon the plant would die. Plants in a very wet environment don't need to worry about losing too much water. Water is plentiful and the air is very humid. Not a lot will evaporate and transpirate out of the leaves, so they have a lot of stomates to maximize photosynthesis and influx of CO2. A plant's stomatal density is influenced then by the CO2 concentration, the temperature, and the sunlight conditions during maturation. If there's a lot of sunlight and high temperatures, lower stomatal density, very low CO2 concentration, not as much sunlight, a lot higher concentration of stomates. They want to maximize CO2 intake, decrease H2O output. So, if you remember, like when we review C4 and CAM plants, evolved specific mechanisms of photosynthesis to limit stomatal opening and closing. So CO2 depletion and sunlight presence stimulates stomata opening at the beginning of the day. So over a long night, when the plant has used up its CO2 for the photosynthetic reaction, CO2 will be low. That'll stimulate the stomates to open in the morning, and also the new presence of sunlight, getting ready to photosynthesize, will stimulate those plants to open their stomates. So here are some stimuli for stomatal opening. Light shining on these guard cells will open the guard cells and allow the stomates to open. And when there is not light, the guard cells will close and the stomates will be closed. Also, high atmospheric for CO2, high atmospheric concentration of CO2 will create a lower stomatal density, low CO2, high stomatal density. We'll talk about the role of potassium in stomatal opening on Tuesday. Something a little bit more detail I just want to get into after we've had an experiment with our lab tomorrow. Lastly, nutrients move from source to sink. So this is mainly sugars that travel through phloem. Sugars travel from areas of the plant that make them via photosynthesis, so like leaves, so sources to places that use them, sinks, like roots. The phloem transports sugar from sources to sink. So positive pressure is responsible for the movement of sugar from source to sink. So if this is a sucrose source, maybe like a leaf, and this is a storage root, a sink, the concentration gradient will cause positive pressure for that sucrose to flow from its source to the sink. All that we need to make sure is that the sinks use up this sucrose fast enough to always have a lower concentration of sucrose than the sources. Sugar at high concentrations and in areas of the plant where it is made, and at low concentrations in the area where it's utilized. So sugar is loading these green dots into the seed tubes of the phloem, and it reduces this water potential inside the seed tube, and this causes the tube to take up water by osmosis. So the sucrose goes into this phloem, it increases its concentration, which causes the transport here of water into the phloem tube. The uptake of water generates a positive pressure that then forces the sap to flow along the tube. So flowing from source to sink, there's not a lot of sugar. The pressure is then relieved by this sugar unloading into the sink, where a lower concentration. And if there's an unloading, all of a sudden this concentration of sugar in the phloem is going to drop, causing water to move back over to the xylem. So we're cycling water from xylem to phloem to aid in the flow of sucrose from source to sink. Okay, that is it for our video for today, and I will see you in class.